During a prolonged fast, the body enters severe biological rationing. It must secure enough energy to sustain the brain's relentless, unyielding demand for fuel. This timeline tracks primary energy reserves. By the 24-hour mark, hepatic glycogen crashes to absolute zero. Over the next 2 days, free fatty acids and ketone bodies show an exponential rise. Yet despite the complete absence of glycogen, circulating blood glucose remains perfectly flat. To prevent the brain from starving after day 1, the organism deploys a secondary metabolic engine that dynamically substitutes for glucose. When glycogen fails, the liver immediately defaults to gluconeogenesis, the synthesis of new glucose from non-carbohydrate sources. To build these new glucose molecules, the liver requires a steady supply of raw materials. Specifically, it needs carbon skeletons derived from glucogenic amino acids. Harvesting these amino acids exacts a massive physiological toll. The body must actively dismantle its own functional muscle and structural proteins to secure the carbon needed to keep the brain alive. To avoid this fatal self-cannibalization, the organism must shift its systemic reliance away from glucose entirely, turning instead to its massive stores of hydrophobic triacylglycerols. Surviving starvation requires solving a complex logistical problem. The body must extract hydrophobic fat and convert it into a water-soluble fuel the brain can accept, thereby sparing critical muscle tissue. This shift begins in the adipocytes. At the onset of fasting, the hormonal landscape shifts. Insulin levels drop critically, while glucagon and epinephrine rise. This drop in insulin directly activates a highly specific regulatory enzyme located within the fat tissue, hormone-sensitive lipase, or HSL. This schematic shows a triacylglycerol molecule under attack. HSL cleaves it into 3 fatty acids and 1 glycerol. The isolated glycerol returns to the liver for gluconeogenesis. The hydrophobic fatty acids cannot dissolve in blood, so they bind to albumin to travel safely. Lipid mobilization unspools massive, inert energy reserves into the bloodstream, but these raw fats cannot supply the brain yet. They require heavy chemical processing. That processing occurs inside the hepatic mitochondria. The breakdown of fatty acids is strictly compartmentalized, isolating it geographically from lipid synthesis. Long-chain fatty acids cannot diffuse through the inner mitochondrial membrane on their own. They must be escorted across the border via the carnitine shuttle. This flowchart illustrates the 4-step loop of beta oxidation: oxidation, hydration, a second oxidation, and cleavage. It's the precise chemical reverse of fatty acid synthesis, breaking the molecule down. A single cycle yields a 2-carbon acetyl-CoA block, plus the reducing equivalents NADH and FADH2. The NADH and FADH2 are immediately utilized by the electron transport chain, generating massive amounts of ATP to power the liver. Beta oxidation functions as an industrial shredder, converting long fat chains into pure cellular electricity and raw acetyl-CoA fragments. There is one singular exception to the even-numbered rules of beta oxidation: odd-chain fatty acids. When shredded, they leave behind a 3-carbon molecule called propionyl-CoA. Unlike the 2-carbon even-chain fragments, this 3-carbon odd-chain fragment can cross over to enter the gluconeogenesis pathway. Processing this fragment requires the mitochondrial enzyme PCC. A defect in this enzyme results in a severe clinical disorder known as propionyl acidemia. This bar chart displays an experimental attempt to rescue this defective enzyme using peptide fusions. The data tracks the production of the molecule MM-CoA across deficient cells. The results show that only the constructs equipped with a specific mitochondrial targeting sequence successfully restored metabolic function. This experimental finding reinforces normal cellular operations. The physical barriers between the cytoplasm and the mitochondria dictate strict metabolic control. Mitochondrial isolation ensures that enzymes are entirely useless unless they are deliberately smuggled into their correct subcellular zone. Returning to the main beta oxidation pathway, the rapid shredding of even-chain fatty acids results in a massive pileup of acetyl-CoA units. At this exact moment, the citric acid cycle grinds to a halt. It is negatively inhibited by the high levels of ATP and NADH freshly produced by beta oxidation. Here we can visualize the bottleneck. Acetyl-CoA units pour into the hepatic mitochondria, but the primary mechanism for burning them is jammed shut. This accumulation triggers secondary allosteric effects. The excess acetyl-CoA signals the inhibition of glycolysis and pushes remaining pyruvate into gluconeogenesis. The liver is drowning in high-energy acetyl-CoA, but because acetyl-CoA cannot cross the blood-brain barrier, the brain itself is still starving. The liver solves this crisis through ketogenesis, utilizing a specialized relief valve designed to clear the acetyl-CoA bottleneck. In this mitochondrial sequence, the liver condenses two acetyl-CoA molecules to form the larger intermediate HMG-CoA. The immediate cleavage of HMG-CoA produces acetoacetate, the first true ketone body. Through a subsequent reduction, a portion of acetoacetate is converted into the more stable 3-hydroxybutyrate. These new molecules possess a crucial physical property. Unlike raw fatty acids, ketone bodies are highly water-soluble and require no carrier proteins to travel through the blood. Ketogenesis successfully transforms trapped hydrophobic energy into a universal, easily transportable fuel designed specifically for the brain. Because of their solubility, these freely floating ketone bodies travel unobstructed through the bloodstream and easily cross the blood-brain barrier. Once inside the mitochondria of brain and muscle cells, an inverse process takes place, ketolysis. Reactivating acetoacetate back into usable acetyl-CoA requires an essential enzyme known as thiophorase. Looking at this tissue map, we see a brilliant anatomical constraint. The liver contains every enzyme to produce ketones, but completely lacks thiophorase to burn them. This absence prevents the liver from cannibalizing its own fuel, forcing ketone bodies out to peripheral tissues. This enzymatic blind spot guarantees the brain receives the absolute priority of the organism's energy supply during a prolonged fast. Once starvation extends past a week, the brain actively metabolizes ketone bodies for up to 2/3 of its total energy output. This creates a systemic physiological domino effect. As the brain utilizes ketones, it stops demanding new glucose, signaling the liver directly, halting gluconeogenesis entirely. With gluconeogenesis shut down, the muscles receive the final instruction. They no longer need to harvest their own amino acids for carbon skeletons. Lipid mobilization and ketogenesis represent a unified, multi-organ firewall. They evolved specifically to protect human muscle and structural integrity against the fatal threat of starvation.